Good morning, everyone, and welcome to Elixir Friday Coffee, where we present the tools and services provided by Elixir Czech Republic, which is infrastructure for biological data. This time, we have a speaker from the Biological Center in České Budějovice, Petr Novák, and he will tell about the RepeatExplorer, which is a tool for genome assembly and annotation. So, Petr, floor is yours.
Okay. Thank you, Jirko. So today I will talk about some specific tools we have on our RepeatExplorer server. This server is provided by our group and in collaboration with Elixir Czech Republic and CERT Scientific Cloud and CEST. And I will talk about like two pipelines. One is for annotation of transposable elements, and the other is called the repeat annotation pipeline, which is more complex. The server can be found on this address if anybody wants to explore these tools. And before I will start talking about the genome assembly annotation, usually you can find in the literature term repeat masking. So people who are interested in genome annotation, who are interested in the genes, they just use repeat masking because they want to get rid of these repetitive elements from the assembly because it is interfering, or it can interfere with the annotation of the genes. On the other side, our group is interested specifically in the repeats. So if we are annotating genome, we actually want to identify and annotate these repeats. So we distinguish, or two big groups of repeats are distinguished: interspace repeats and satellites repeat with a tandem organization. Other types of the repetitive DNA you can find could be rDNA, and they often correlate with the formation of heterochromatin. The repetitive sequences has a function often like in making structural features of centromere. So if you look at the tools which could be used for repetitive sequence annotation, you basically need to distinguish several categories. So if you have a complete genome assembly or partial genome assembly, and if you have your library of repeats, you can use one group of tools. One famous group tool is a repeat masker. Then our tools, which we are providing on the server, are actually more oriented on the cases where you don't have an assembly and you don't have a library of repeats. So it is so-called a clustering-based approach, and this approach is using raw sequence reads and to obtain the information about the repetitive sequences in the genome. If you want to annotate any type of the repetitive DNA, you usually work with some reference databases. One known database is ReBase, which is now commercial. There is a DFAM database, which is an open database, and on our server we are providing RexDB database, which is a database of protein domains which are coded by transposable elements. Very often, these databases contain information about the repetitive sequences from a model species. So if you do not work with the model species, sometimes these databases are not enough for you. Now, on the server, we have some tools. Mostly we were using and promoting these assembly-free tools. So this is a RepeatExplorer tool, and Tarian and ChIP-seq mapper. All these tools assume that you don't have a assembly and you want to annotate repetitive sequences in your genome of interest. But we have also tools which can be used directly if you already have a genome assembly. So these are used. So this is a tool Dante, and then we have a repeat annotation pipeline. The advantage of the assembly-free method is obviously that because you don't need this assembly, it's usually enough if you have just a fraction of the genome sequenced. So this characterization is basically much cheaper. And then, but the abundance of these repeats could be affected by the sequencing technology, by the bias from the sequencing technology. On the other side, here, when you have a genome assembly, then you are also affected by some incompleteness of genomic assemblies. So first, I will talk about the tool Dante. This tool can be used on basically any genome. You don't need any additional information, and it is mostly suitable to use it on Viridiplante species. And this database contains protein coding domains of transposable elements. And we have also a version which is oriented for which covers a metazoa species, but this is less comprehensive. This database basically is composed of the proteins. So here you can see on the left side, you can see the scheme of the transposable elements which are present in the RexDB database, and these colored areas are coding sequences. These are conserved domains, and these are actually part of the RexDB database, and they are classified based on the phylogenetic principle. Usually, if you look into other reference database of the repetitive of transposable elements, you see some classification of these transposable elements, but this is usually at the level of superfamilies. In the database, with the RexDB database, we go actually further, and because it is using this phylogenetic principle, we are classifying repeats into lineages based on the phylogeny. And the advantage is also that we have some reproducible categories, and which could be also used for some interspecific comparison. So the principle of Dante tool is quite simple. You have a RexDB database, and this is used for scanning and finding similarities in your genome. We record basically multiple hits if they are found for some specific regions, and the classification of these hits is evaluated. This threshold is dynamic, and it is based on the highest score of the best hit found. And at the end, we evaluated the hierarchical classification of these hits, and you end up either with some clear annotation when we can annotate a region and a protein domain into the specific lineage, or in case we found some conflicts in the annotations in the best hits, then the lowest common ancestor is used for a classification. When you perform this annotation, then you will obtain these results in GFF3 files, and you will also get extracted protein domain sequences. So here you can see the multiple protein domains from single transposable element lineage. In the genome browser, the advantage of having a tool on the Galaxy server is that it can be directly visualized using a JBrowse genomic viewer. Or alternatively, you can download these results and use them for the further analysis. So what are the applications of these? There's one application, and it's actually phylogenetic analysis of transposable elements, and these results can be also used for dating of a transposable elements activity. There is a classical methods which use for dating of transposition actually the information from the inserted retrotransposome. So here you see the scheme of the transposable element, and the boundary of it is formed by a long terminal repeat. At the time of the transposition, this sequence of the long terminal repeat is they're identical. But during a time, the mutation accumulates, and the one method which is trying to date the time of the insertion is actually working with the comparing these two LTR sequences. And more mutation you found, then the older element it is. So this can be used for the dating of these retrotransposition. However, in the real genome, it's more complex because you often have transposable elements which are not complete. There are always some new transpositions. You have a nested insertion, and it's become difficult to identify the corresponding pairs of the LTR sequences. So, and if you don't have a correct pairs of the LTR sequences, you don't get correct results of the dating. So the Dante basically can be used to calculate the dating of the retrotransposition in different way, just based on the phylogeny. So basically, you can use some conserved domain, either most often RT domain or integrase domains, and these could be used to create a phylogenetic tree. And based on the analysis of the branch length of these trees, you can estimate some branching time. So here is the example of trees of phylogenetic trees for two elements in the Pisum sativum genome, in the garden pea genome. And here you can see the retrotransposome TI1 copia, Angela, and you can see there are a lot of short branches. And this mean that there was a recent wave of amplifications. While on this retrotransposome here, you can see that the branches are much longer, and there wasn't really any clear wave of amplification. So these histograms are summarizing the lengths of these branches. So this is one application how Dante can be used. Additionally, because we are distinguishing transposable elements to the lineages, not only superfamilies, you can use this information to evaluate the targeting of individual lineages of transposable elements in the genome. So here you see the what's the density of the gene along the chromosome in the Pisum sativum genome. This arrow here shows the position of the centromere. And so you can see that there are low density of genes around the centromere and the higher at the chromosome arms. And if you look at the superfamily distribution along the chromosome, which is copia elements and gypsy elements, so you can see that this is correlated or negatively correlated with the centromere position. But this category is actually quite broad, so we can break it using Dante pipeline to lineages. And here, for example, you can see that there are several types of the gypsy elements. One is a TK, which is then retant, and you can see that they're targeting in the on the retrotransposition is different. So TK is more targeting a centromeric region, while retant is targeting whole genome evenly. And here, this is a centromeric chromovirus, and you can see that here is a clear targeting to centromeric region. So Dante analysis enabled us to analyze LTR retrotransposon in more specific way. So this was a Dante, and now I will go to the second pipeline. It's called RepeatExplorer pipeline, and so again, if you look at this table, when we have a divided repeat annotation method to the groups, so if we have some species for which we do not have a good reference database, we need to create some custom reference database. This custom database could be created either from this genome assembly. If we have a genome assembly, we can use tools like Repeat Scout or Repeat Modeler to create a custom reference database. Or we can actually use our RepeatExplorer 2 pipeline or Tarian pipeline and use a raw sequencing read to create such a sequence database. The advantage of using this approach, using a RepeatExplorer 2, is that very often many repetitive sequences are underrepresented in the genome assemblies, or they could be even missing. And if they are missing, you cannot actually create a good custom reference library of repetitive sequences. So this approach, where we are using basically primary data which were used for creating the genome assembly, is can potentially create a library which is more representative. So we are basically using the combination in the RepeatEx annotation pipeline combination of this assembly-free approach, and then to use this library to create a library of repeats and to create a repeat annotation of the whole genome. Before I will go to the whole pipeline, I will just briefly mention the principle of this RepeatExplorer pipeline. So at the beginning, you are using some shotgun genomic sequences with a low coverage, which is less than 1x usually. And because repetitive sequences, which are here represented as these colored blocks, and the reads are black arrows. So reads, if you do a all-to-all sequence comparison, then you can identify clusters, and these clusters correspond to families of repetitive sequences. And then from these clusters, we can create some consensus or partial consensus of these repetitive sequences. So we are analyzing here repeats as clusters. And this analysis in RepeatExplorer is so-called cluster-centered because we are trying to gather as much as possible information about the individual clusters. One way what we do is, again, we use graph representation of the clusters. So this is a so-called assembly graph, which corresponds to overlap layout assembly graph. The vertices correspond to sequence reads, and each read can be compared to our databases. One of these databases, again, RexDB database, and then we have some built-in RepeatExplorer databases which contain, for example, information about the rDNA. And when we compare these sequence reads to these databases, we can identify the similarity. These are here highlighted as colors. And the whole cluster is annotated based on these positive hits. So even the reads for which you do not find any similarity to the databases are annotated as a sum repeat because they are actually part of this cluster. So this is used mostly for annotation of interspersed repeats. And then we have a part which is called Tarian. This is a tandem repeat annotation. And tandemly organized sequences has some specific, create graphs with just some specific properties. Namely, you can see this kind of circularity. So Tarian is detecting these kind of structures in the graphs, and these structures are then further analyzed using K-mer approach and De Bruijn graphs, and we will get consensus sequences or some variants. And at the end, each cluster is assigned into into classification, into the node of the classification hierarchy of the RepeatExplorer. And this is basically making our custom library of repeats. So if you want to use a whole repeat annotation pipeline, you would have to start with some shotgun sequencing at the beginning, which we think is better because it's less bias than if you will start to create this library based on the assembly. So you will start usually with the per end Illumina sequence reads, or alternatively you can use a PacBio hi-fi sequence reads with a low coverage. Usually, if you have already assembly, you probably also have data like this, which could be used to create your custom library. Then you will run a RepeatExplorer with a Tarian. If you are interested into how to actually run the whole pipeline, we have a tutorial which is available on the RepeatExplorer YouTube channel, so you can watch this. And at the end, you will get clusters of the sequence reads, and these are basically the collection of the contigs. These contigs contain basically all possible variants of these repetitive sequences. So if you look, for example, in this graph here, this represent transposable elements, and you can see that some part of the sequences are represented by rather like single path, and these are the sequences which are not variable too much. And then you have some parts which are variable a lot, because you can see a lot of like parallel path, and all these paths are represented in the custom database as a collection of contigs. Additionally, second part is like consensus from the Tarian pipeline. This is capturing the variability of the tandem repeats. It is important to know that that tandem repeats are also the ones which are very often missing in the sequence in the genome assemblies, and they are often also species specific. So you cannot easily use some model species repeat libraries to detect these. You need to use some like a feature-based approach to detect the tandem repeats. And all these clusters are annotated, and what we really highly recommend that you will do some manual curation of your results, because you cannot take the annotation for granted. There are a lot of exceptions in the biology, so it's always good to explore these annotation critically. And again, it's a part of the tutorials on the RepeatExplorer YouTube channel, so you can watch these. And at the end, you can, of course, again, visualize your results directly on the Galaxy server in the JBrowse genome viewer. So here you can see, you can visualize together Dante results and repeat annotation pipeline results. So here you can see the results from the Dante, which are just protein domains. And here you can see the whole element annotated in the genome. Okay, so this is how repeat annotation pipeline is used. It's quite simple and straightforward. And I will just show some application how this pipeline and annotation of repeats can help us.
I am from the group where we that are like long-term model or cytogenetic model is a Pisum sativum, garden pea, and garden pea genome is exceptional in the term that it contains a huge number of the satellite sequences. Here you can see them as these colored dots. And these satellite sequences are very diverse. And another exceptional feature of the Pisum sativum genome is that it has a huge centromeres. These centromeres are as big, a single centromere could be as big as a complete Arabidopsis thaliana genome. And because we are interested in the structure of these centromeres, we know from the wet lab experiment, specifically for cytogenetic experiments, where you can see that you can use a fluorescence hybridization in situ to identify individual satellite sequences. These satellite sequences, and a lot of them were identified using RepeatExplorer and Annotation Pipeline, and they could be actually mapped to the centromere. So this is a centromere here, and you can see that each chromosome has actually different centromeres, and there are multiple different types of the satellite sequences in the genome. And our group was a part of the genomic consortium, and there was a first assembly of Pisum sativum published 2 years ago, and while this assembly was quite complete, it was really depleted for high copy repeats, and centromeres were quite poorly represented. So if you look at this result, so this was obtained also from using this repeat annotation pipeline. So we can evaluate the abundance of the repetitive sequences in the genome either using RepeatExplorer and the raw data. So this is shown here on the x-axis, or we can use this library of the repeats to annotate the genome assembly. So this first version of the genome assembly, which was annotated by RepeatExplorer pipeline, by repeat annotation pipeline. And here you can see if what is the representation of these individual repeats in the genome assembly. And what you can see here, these green ones are satellites. And you can see they are really highly underrepresented in the genome assembly. Then rDNA is highly underrepresented. Other repeats are represented a little bit better. And so we can basically see from that centromere sequences couldn't really be possibly complete in this assembly. So recently, the second version of the Pisum assembly was started to be generated, and here you can see the comparison. So this is again this Pisum sativum version 1, where you can see this underrepresentation of satellite sequences. And here 3 other panels represent the alternative assemblies or drafts of the assemblies. One was created using Oxford nanopore reads, and 2 of them were created using PacBio hi-fi reads. And you can see that there is a great improvement in the representation of the satellite sequences, especially in this hi-fi PacBio reads. There is also improvement in the Fly Oxford nanopore. So this repeat annotation pipeline can be actually used as some quality control measure of your genome completeness. Usually people use completeness of the gene coding sequences. For example, Busco method is used to evaluate the completeness of single copy orthologs. But because we are interested in repeats, this criteria for us would be much better. So from this we know that it's could be possible to make a centromeric assembly of this centromeric region more complete. And indeed, this is an example of centromere from the chromosome 6, which was assembled by Jiří Macás. And so on the top panel, you actually see the annotation, bioinformatic annotation of this centromeric region. Each color is a different type of satellite, which was annotated by this repeat annotation pipeline. And the below this picture here represents actually the results of the fish mapping in the web lab, which was done by our postdoc Laura Avila Robledillo. And so here you can see that on the packetine chromosome, you can actually use these sequences which were obtained from repeat annotation, use them as a probe, and you can actually verify that the assembly, that the order of the satellites in the assembly is correct and is the same like the order you can actually detect on the packetine chromosome. Okay, so this was the one of the application. This work was done in the laboratory of molecular cytogenetics. Head of the laboratory is Jiří Macás, and Pavel Neumann, Laura Avila Robledillo, Andrea Koblíšková, and Nina Hošťáková were working on either on the wet lab or also programming the pipeline. And then I also want to thanks to people from Masaryk University, especially Zdeněk Salvet, Ivana Křenková, and Martin Macháč, who are taking care of our RepeatExplorer server, and also they are doing some user support. And if you want to see how practically this pipeline is used, I can recommend you to go to the workshop which is organized by Martin Czech next week. Okay, thanks for your attention.
Thank you, Petr. We have a few questions here. I don't know at which level of the expertise are they. So my first question is, what precisely does the REXDB contain? Transposable elements in form of protein domains?
Yeah. So they contain protein domains which are coded by transposable elements, and this was a manually curated database which contained thousands of complete elements. So we know that are possibly functional elements, and these protein domains are also defined, so they have actually the same boundaries, and they could be easily used for phylogenetic analysis because it's like they could be compared directly.
Mm-hmm. Akara is asking, how do you define lineages, phylogenetically or by some specific mutations?
Yeah, well, these are defined phylogenetically as a clade on the tree. There are still tendencies to define the families as for example, one definition, which old definition is defining the families as a sequence which has at least 80% identity over 80% length of the element over at least 80 base pairs. But this definition kind of doesn't work always, and I think it doesn't really have a good foundation in the phylogenetic. So we use a phylogenetic definition.
Mm-hmm. Okay, I think the last question, which I think is a bit uneducated, at which extent are the repeats a noise, and at which extent do they contain important regulatory or functional elements? Can you say what is generally their major role?
Well, obviously they could be viewed as a noise, but they are still shaping a genome. They can inflate the genome, and also, but the genome, the mechanism in the genome also wants to try to get rid of them. So there is a dynamic, and very often there are examples. So it's like evolutionary force of the genome, and there are examples of so-called exaptation where regulatory element or some regulatory gene elements are actually derived from the repetitive elements. and then they form clearly some important structural features like centromere or for example telomeric sequences.
And at which extent are they silent?
Well, they are not too active, right? But they apparently were, they could be some stress response when they they could be activated, for example, by a drought. So they're like thinking that they can actually force the evolution of the genome, but most of the time they're not too active. But using this phylogenetic analysis, you can actually detect that in the history there were some bursts of retrotransposition.
Okay, so because there is no other question, Petr, thank you very much again, and have a good day to all of you, and see you next Friday. Bye-bye.
Bye.